let's start the second part of our symposium. So good morning. I'm Maria Paula, PhD student from the Bioenergy program at Unicamp. And I'm delighted to introduce my fellow colleagues in their project speech presentation. Understanding the importance of communicating our research in a clear and concise manner, our fellow colleagues agreed to briefly explain their work to us. Students enrolled in our program from all three universities will have 3 minutes to share their projects in an informal manner. So please welcome them. Our first guest is Bruna Scaramboni from Unesp. Bruna.
Good morning, everyone. So, how can we improve glucose and ethanol production in Brazil? This work, we studied the improvement on enzymatic cassava bagasse treatment to obtain glucose and ethanol biosynthesis. I'm a third-year PhD student from Bioenergy program, and my advisor is Professor Pedro de Oliva Neto, and this study was developed in Bioenergy Research Institute at Unesp Campus Assis. So our question was, could cassava bagasse be included in a renewable platform for the production of ethanol? And some numbers to understand our issue is that Brazil produced 23 million tons of cassava in 2014, and this can generate around 23 tons of cassava bagasse too. In dry basis, 3.3 million of tons. And so, as an example, near Assis we have a factory of Terio's company, and driving on the highway, it's possible to see a large mountain of cassava bagasse. So what to do with that? In our study, we propose 3 steps for treat the cassava bagasse. So, first, amylase production in solid-state fermentation in optimized conditions. After that, we use cassava bagasse and enzymatic hydrolysis of cassava bagasse using fungal amylase produced here and commercial cellulase. And the best proportion was defined in using the experimental design tool. And after hydrolysis, we obtained glucose, and glucose was fermented to ethanol. So we achieved 61% of conversion, cassava bagasse into sugar. And here, maybe it's a little difficult to see, but here is cassava bagasse treated with amylase and cellulase, and here without treatment. Thank you, Michel Brienzo, for the microscopy. And about ethanol production, with this study, we have the potential of produce more than 200 liters ethanol per ton of cassava bagasse in dry basis. And considering all cassava production in Brazil, using this bioprocess, it would be possible to increase the 3% ethanol production in Brazil, the second largest ethanol producer in the world. So without food competition. So this is just to show you. This is a previous study accepted for publication about the same area too. And thank you very much for your attention.
Before introducing our next guest, I would like to give a quick announcement. Those speeches will not have questions, so I encourage you to question them during brunch, which will be served the same place we had our coffee break, okay? So our next guest is Fernando César Barbosa from Unicamp.
Hello everyone, I'm Fernando, and I will present to you a little of my PhD project that will be developed here at Unicamp under the supervision of Professor Rosana Goldbeck, entitled as Optimization of Cellulose-Saccharide Production by Enzymatic Hydrolysis of Sugarcane Straw via Heterologous Expression of Cellulolytic and Oxidative Enzymes. Okay. So first of all, I'll give you a little overview about where my project is inserted in. So I'm working on a thematic project of a PESP in collaboration with researchers from UK. And the main goal of this thematic project is to produce ethanol and make the biodigestion of vinasse from sugarcane straw and forest residue. And I'm working here in the specific part, of the project responsible for enzymatic hydrolysis of already pre-treated by hydrothermal and diluted acid sugarcane straw. And my goal is to produce cellulosic saccharides for further fermentation for other groups from the thematic project. Okay. So why we wanted to use cellulosic saccharides? That's because the cellulosic saccharides give to the process some advantages, such as lower process contaminants, shorter total fermentation time, high binding affinity to cellulose substrates, and it does not limit the process by high glucose concentrations. So that's the advantage to use the cellulosic saccharides in spite of glucose. And how we wanted to make that? So first of all, you make a characterization of pre-treated sugar cane straw that you receive. So you measure the concentration of cellulose, hemicellulose, lignin, and ashes, and then you work in the enzymes production. You work with endoglucanase, LPMO, that are oxidative enzymes, and CDH, that's cellulobiose dehydrogenase enzymes. And you already make the literature review to identify the key enzymes, and then you make the selection from promising genes. You clone the selected genes in E. coli, and then you produce and purify these superexpressed enzymes in E. coli. After that, we will measure the enzymatic activity of these enzymes, and then we will evaluate the key enzyme mixture by experimental design to maximize the cellulosaccharides production. So we use the design plaquette burner to identify the most important parameters and enzymes in macro scale. And our goal is to develop a mixture of enzymes able to optimize the cellulosaccharides production. Okay? And then with that, we wanted to produce a hydrolyzed broth rich in cellulosaccharides for further fermentation for other groups from the thematic project. And last, as a plus of my PhD project, we are planning to make a sandwich to engineer Saccharomyces cerevisiae in order to produce ethanol directly from cellulosaccharides, that it doesn't happen at the moment. Okay? So that's it. Thank you.
Now, Jorge Eduardo Cardona Flores from Unicamp will talk about his project.
Hello, everyone. My name is Edward. My advisor is Professor Antonio Rull from Physics Institute. So I'm going to tell you a little bit about my project. My research project is related to enzymatic biofuel cell. But what is this? Enzymatic biofuel cell is a device capable to transform chemical energy into electricity. Similar to conventional fuel cell, enzymatic fuel cell consists in two electrodes, an anode where fuel is oxidized and delivering electrons by generating electricity. On the other hand, a cathode when generally oxygen is reduced. The principal difference between enzymatic fuel cell and conventional fuel cell is just the catalyst in enzymatic fuel cell. We are using enzymes for catalyze the reaction, and a conventional fuel cell uses platinum-based catalyst. What are the current problems with this technology? The principal problem is just related with immobilization of the enzyme on the electrodes, because the enzyme can suffer structural changes during the process of immobilization. These structural changes resulting in short lifetime of the biofuel cell. And the other problem is the electron transfer between enzyme and the electrodes. So the major technical drawback of enzymatic biofuel cells is finding feasible design of electrode. Our proposal is I'm going to explore different materials, principal graphene-based materials and polymers with desirable electrical properties to construct electrodes. How? We are going to use techniques such as LBL, layer by layer techniques. This is a method of physical absorption on surfaces, so you can construct 3D structures with different materials, so you can meet in different properties of the different materials in only one structure. The idea is this. Thank you.
Now, I'd like to present to you Marijke Wijma from USP.
Which one do I point? Good morning, everyone. My name is Marijke Wijma. I am Professor Glaucia Mendes Souza's student, and I'm from the University of São Paulo in São Paulo. Okay, so I would like to discuss just an overview of my project and what we do in our lab there in São Paulo in the Chemistry Institute. So we try to study sugarcane on a molecular level by looking at the transcriptomic and metabolomic analysis of this organism. So I study a specific variety. It's the SP803280. I don't know if you people know this, but I think most people within the sugarcane industry know this variety. So the main objective is to study the sugarcane variety on a morphological, physiological, as well as molecular level. We look at the gene expression as well as the metabolite levels and how this changes throughout growth and development, to try to understand what governs and what regulates sugarcane growth within the field. So just a quick overview of the experimental design is two field experiments were conducted. One was initiated under low average temperature and rainfall, and the second one was initiated over high initial temperature and rainfall. We would like to see how this environmental factors affect the growth and the development in the field. So we collected samples of the leaves, the immature, the young, as well as the mature internodes. And then we conduct in-field experiments by looking at the morphology, such as the height, the number of internodes, etc. We also conduct biochemical analysis like the bricks content, the different pigments, and how all of this changes within the field. And then the part that I'm working on is the metabolomics part, where I try to identify and see how the different metabolites change within this organism throughout the growth and development. So we have a pipeline in our lab that we use to process and analyze the data, but this is the major drawback when you work with liquid chromatography mass spectrometry. So if there is someone that wants to talk to me during lunch about this part, I would like to get to know how to do this more in-depthly. And then also we work with transcriptomic data, mainly with microarrays, and then we have our own online database tool where we have genomics, transcriptomics, as well as metabolomics data that we analyze. And the idea is to try to integrate all of this to understand exactly what happens within the organism throughout growth and development and how we can improve this organism further. So this is an example of what you would expect. I use MetaboAnalyst, so I get the metabolite levels, normalize them, do statistical tests, for example, find the significant metabolites, and then I try to correlate it with different transcript levels within this organism. And that is it. So if everyone wants to talk to me afterwards, not about this, but about this, you can. Thank you.
Now we're going to have Matheus Eugênio Boscaro from Unesp presenting his page.
Morning, everyone. Well, when Richard Feynman, the physicist, talked for the first time about nanotechnology, we couldn't imagine how big it will become. Today, the nanotechnology plays an important part in many areas, many scientific areas, including the bioenergy sector. And what I am doing in my PhD is trying to use iron oxide nanoparticles to improve the production of biogas in anaerobic bioreactors. This nanoparticle is magnetite nanoparticles, and how can these nanoparticles improve the production of biogas? Well, it can be used to supply iron to the main, and also can use as a catalyst or changing the redox balance of the main. So the main objective of my work is to produce these nanoparticles and use this in a specific way to enhance the production of biogas. And the first challenge for this is to produce the nanoparticles. I am using the co-precipitation method. Here we have some steps for the production. We use a mixture of iron ions and a solution of alkali to produce some magnetite nanoparticles. Here we have a previous analysis to characterize the material. Here we have the UVV spectra, and here we can see the iron band, and here a small band showing us that the particle is really a nanoparticle. And we are trying to apply all of these particles in the next step of the work, so I don't have many results today, but this is the idea. Thank you.
Now Regiane, please. Regiane Alves de Oliveira. She's a student from Unicamp.
So good morning, everyone. My name is Regiane. I'm a student from Unicamp. My advisor is Professor Rubens Maciel Filho, and I worked in partnership with Professor Carlos Eduardo Vaz Rossell and Dr. Joachim Venus from Germany. And my project was about producing first and second generation lactic acid from sugarcane. And I also worked a bit in Germany, producing lactic acid from another lignocellulosic material using cellular cycle fermentation of Bacillus coagulans. So the first question is why to produce lactic acid? Because we have many applications for this, and it has a high value. And nowadays we are producing lactic acid from processed sugars, and this is competing with food, basically. And the main applications of lactic acid are nowadays for food, pharmaceutical, and cosmetic industries. But we also can produce almost all of the plastics produced from oil with the same properties from lactic acid. We also can produce the biodegradable plastics that are very good for the environment nowadays. Lactic acid also is a building block molecule that you can convert in different chemicals. And the main importance, the main focus today is to produce polymers for replacement of titanium processes in the medical area. So what I did basically was using fermentation methodology to produce lactic acid. I chose 2 different bacteria, Lactobacillus plantarum and Bacillus coagulans, and 3 different substrates to produce the first and second generation lactic acid. I worked with molasses from sugarcane, the sugarcane bagasse after the hydrolysis, the pretreatment and hydrolysis, enzymatic hydrolysis, and also sugar beet pulp after pretreatment and enzymatic hydrolysis. These 3 substrates have different compositions, and they had totally different profiles of lactic acid production. And the conclusions I have until now are that the production of lactic acid has high yields, high productivity, and you can reach high titers. I could reach at the maximum 250 grams per liter of lactic acid in batch fermentations, and the conversion is more or less around 97%. The sugarcane molasses and the cellulosic and hemicellulosic hydrolysates are very good substrates for producing lactic acid, and also the sugar beet pulp that I used in continuous mode of fermentation. Yeast extract is an important component, but just for the sugarcane substrates, because in the case of the sugar beet pulp, the biomass has already a lot of proteins and amino acids, so I didn't need to use yeast extract. As we have a lot of sugarcane in Brazil, the use of yeast extract wouldn't be a problem, as we have the yeast is produced from ethanol industries. So for the Brazilian scenario, it's still okay. I also tested different bases to neutralize the acid produced, and I had 2 different profiles of production with sodium hydroxide and calcium hydroxide. And the 2 microorganisms I choose have the ability of consuming the sugars of cellulosic and hemicellulosic substrates without detoxification. So they actually have a very good performance using some of the toxical components of the hydrolysates. And for the downstream process, I tested some different methodologies, and the best methodology that I had the best results was using electrodialysis. And that's it. Thank you.
Please welcome Renato Cruz Neves from Unicamp.
Hi, everyone. I'm going to give you an overview about my work. And I'm Renato Cruz Neves, currently enrolled at the PhD program in Bioenergy. I work in Brazilian Bioethanol Science and Technology Laboratory. And when we think about bioethanol, you think about the integration of the second generation sugarcane biorefinery to produce more ethanol and sugar, this integration into the first generation sugarcane biorefinery. Instead of using sugarcane bagasse and straw to produce more ethanol and sugar, you can diversify your bases, producing biojet fuel through a thermochemical route. This is the main goal of my PhD, integrate the thermochemical route to produce biojet fuel and the first generation sugarcane biorefinery. The point is here? Yes. Okay. So, as a way of trying to explain about how this integration is performed here, you can see the sugarcane biorefinery, the green box, producing ethanol and sugar coming from sugarcane and straw recovered from the field. And then you have a surplus bagasse mixture with straw. We are calling it LSB, which is a lignocellulosic biomass. It's a mixture of surplus bagasse and straw. And then it goes to the pyrolysis process to produce biochar and bio-oil. This mixture is known by bioslurry from this material. And then this bioslurry is gasified in a gasification process to produce syngas, and after gas conditioning and cleaning, you can convert it to syngas and biofuels through Fischer-Tropsch synthesis. You can also produce more green diesel, green gasoline, and electricity. The battery is low? Okay. And in order to understand a little bit about more pyrolysis process, I also performed, I also performed fast pyrolysis in experimental scope. Here you can find the experimental scope of lignocellulosic biomass coming from first generation sugarcane in comparison with simulation data considering kinetic model and yield model. As you may see, the results simulated here are with a good agreement with the experimental scope. For example, the bio-oil fraction together with the biochar fraction, both combined are named as bioslurry. And to identify the final product of this integration process between thermochemical route into the first generation route, I'm showing here the biojet fuel and naphtha production mass flow coming from the end of the disintegration process. Here in the blue bar, you can find the biojet fraction produced from Fischer-Tropsch synthesis, and on the red bar, you may see the green naphtha production. Average carbon ratio for biojet fuel is around C12, and whereas for green naphtha is around C8. Remember that biojet fuel is a complex mixture of biocomponents in the range from C8 to C20. It comprises paraffins, olefins, and other compounds. So that's all. Thank you. I would like to thank CBE, this opportunity here. And also KIT I went to the PhD sandwich over there last year, and I performed the fast pyrolysis process. Thank you.
Now we're going to know Rodolfo Silva Martinez's work. He's a student from USP. Welcome.
Hello everyone, thank you very much for this space to present to you the research I'm performing about organic waste to energy in Latin America and the Caribbean, the state-of-the-art literature review. So we are currently carrying out with Professor Alessandro Sanchez Pereira from USP in Sao Paulo. And after realizing that there is a lot of literature already existing in this field, we found out that there is a frame still missing of the state-of-the-art of thermochemical, biochemical, and chemical technologies in the region of Latin America. So organic waste and energy technologies have already been developed and implemented in the region, but still they are not sufficiently managing the waste. They are still not producing enough energy, and they're still missing to meet the greenhouse gas goals. So starting from this, we made a subdivision of what we are focusing on. And regarding the waste, is organic municipal solid waste, agricultural waste, forest waste, and animal dung. So this is basically all the solid waste existing in the region. Then the technologies we are studying are thermochemical, biological, and chemical. Sorry, this is biochemical, it's not biological. So, So the thermochemical is combustion, gasification, pyrolysis, liquefaction. Our co-student was explaining a little bit about these techniques. And biochemical, fermentation, anaerobic digestion, land filling, and chemical is esterification. So a little bit of the strategy we are using for this literature review. So just to give you an overview of what we are doing, is defining the sources is research papers, books, and doctoral thesis. The limitation of scope is a time frame from 2000 to 2018. And the keywords is lignocellulosic, organic or bio-waste, waste or residues, and energy or biogas. So the classification strategy is that after making a list of the databases, we search for the keywords, and then we found the 25 or 30% best articles. Then we combine them, we merge them to select according to the filters we are doing for the region. So we look for the countries in Latin America or the region, and then we do for the subject. So thermochemical, biochemical, or chemical. So far, the preliminary results we have is that organic waste to energy have a good potential to improve waste and energy systems in the region, reducing environmental impacts, along with a series of social and economic benefits. So this is basically second generation technology that, as you know, is the state of the art. So I encourage you to keep on going in this. And if you have any questions, please address to me after the presentation. Thank you very much. Thank you.
Now we have Walter Sevi from Unicamp. No? Is Sergio here? Ah, Sergio is here. Okay, good. So sorry. Let's welcome Sergio Amaral from Unesp.
Controle. Bom, boa tarde a todos. Sérgio Amaral, sou engenheiro da Volkswagen Caminhões e Ônibus, aluno da... Sorry, speaking Portuguese. Because I'm working at the few minutes ago, still working. So, present you about regarding oxidative stability effect at diesel engine with the diesel B10, biodiesel, 30% of biodiesel and 15% of biodiesel. I'm a PhD student, Sérgio Amaral, and my advisor, Mr. José Luis Silveira. Basically, we start working here because of some problems that we have in our engine caused by biodiesel, mixed diesel. That we have here 5% and 7% of biodiesel mixed, and they have a fuel injection problem, fuel system problem, and we stop the trucks. The trucks stopped because at around, for a year, we pay at around 5 million reais for warranty, only for trucks, for new trucks. Okay, it's in warranty time. The beginning of this year, we found out during the test, a validation test of our engines, exhaust recirculation gas, specifically the engine. We check the performance test that we could have around 20% of performance loss. And we started to try to find out which problem, the root problem that caused this situation. Then we remove the fuel injector nose. We send it to a specific laboratory, and we could find a low oxidative stability according to the standards by ANP approval. This vehicle running 2,000 kilometers. Then we change everything. We started to test again for new validation engine. After 1,500 kilometers, again we have this same situation. Then we send to another lab for analysis, and we have the same answer, 8.9 for regarding the test, ANP, the minimum is 20 hours. Then we go to government, start the development of B10, they will release for next year. Then we have agreement for Brazilian government and start the test for B10. And the government are work-based to release in this job for Hansemat is a specific test that we are doing now. We start in February up to now. The first mixed, we start the test like a T0, time 0. We use additives provided by Petrobras, developed by ourselves, and another one for the current market. And we follow the test. And we presenting last month in Brasilia the results showing the problems that regarding a time for storage time for B10 mixed and biodiesel. And we release SOP in March next year. The final report we present in Brasilia next month. So we will test B10. We will do the ability test for two kinds of engines. We send them parts for Germany for test, for evaluation regarding the thermal situation caused by biodiesel in our engine. In also injection after treatment fuel consumption and oxidation oil change interval will be evaluated also. Our target is change together the association, biodiesel association, to increase the oxidative stability in the production of biodiesel, to avoid this situation showed before here. Okay, to increase here, to get more time and avoid the problem regarding warranty. Basically, is our job, sirs. Thank you very much.
Please welcome Walter Seve from Unicamp.
Good morning. All right. This is one of the chapters of my thesis. I'm right in the middle of the PhD in bioenergy. So this chapter calls Spatial Assessment on the Economic Potential of Bioelectricity Production from Sugarcane Straw under these conditions. So we evaluate the 174 mills, operating mills in São Paulo in 2012 crop year. So what we basically did, it was establish a radius around each mill based on their installed capacity. So this yields some good information about the straw availability per hectare, for instance, and also the recovery distance from the mill, from the field to the mill, and vice versa. And also we measured the distance of each mill to the nearest substation from the distribution company able to receive that bioelectricity surpluses from sugarcane straw. So here you have some spatial-based information that's quite important to evaluate the economic results of each case. So here you have the connection per kilometer, you have the installed capacity of each mill, the straw availability, the amount of straw technically able to be recovered, and also the recovery distance from the field to the mill. Based on that, we calculate the bioelectricity production costs. So we establish as the main assumption that the economic viability of the fields would be whether the pixel or whether the mill is lower than $80 per megawatt. As the main results, we have that 118 mills were entirely unavailable to recover, to produce electricity, bioelectricity from sugarcane straw. And we get that 56 mills were partially available, like this map here. So we have areas that are able to recover some straw that could yield this amount of bioelectricity, and we have the black areas that are totally not able to recover that straw. And here are the mills that are partially viable to recover the straw, and in red you have the mills that are not at all. And summarizing the whole state, you have the economic potential of 2.8 terawatts under those conditions that the bioelectricity could be supplied in that crop year. And the technical potential of sugarcane of bioelectricity would be totally unviable based on the on a prior study that we did. In terms of sensitivity analysis, we got that the bioelectricity selling prices are the main parameter that infer in the amount of the economic potential. And also in technical terms, you have the conversion efficiency that it's also quite important to balance that yields different results. And also the discount rate that you use to model the economic structure of the power plant. This is the basic results. We are going to submit the paper for some journals. So if you want to know a little bit more about the work, I'm completely able to answer you. All right, thank you.
Last but not least, let's welcome Wesley Leorisi-Max from Unicamp.
Good morning, it's me again. So now I think I have a lot of time, so you guys had only 3 minutes, I'll use all the rest of the time. I'm kidding. So I am from the first year of this program. I'm now in the final months of my PhD. And here at Unicamp, my supervisor is Andreas. I'm enrolled in this dual degree PhD program between Unicamp and TU Delft, and there my supervisors are Jack Pronk and Tom Vamaris. So I'm working with industrial anaerobic process. Imagine in industry a reactor at this size, gigantic. Now imagine that you need to keep this thing aerated. Your organism will only grow and produce if you have air well mixed inside this gigantic liquid thing. You can imagine that this is gonna take, you're gonna need to spend a lot of money with this. In our reality in Brazil, we produce ethanol just fine, without aeration, anaerobic process. The vessel is even open. The yeast is so good in doing anaerobic process. But imagine if you have a product, for instance, organic acids, like lactic acid, that Regiane talked before, and you want to produce this using an eukaryote, like yeast. You don't want to use bacteria. Sometimes, depending on the process, bacterial fermentation can be expensive. Nutrient, you have to avoid virus infection, and you have to control the pH. So we want to use Saccharomyces cerevisiae yeast. But if you want to produce these molecules using yeast, the problem is that they don't export this like ethanol, just without expending energy, without expending ATP. To export these molecules, lactic acid, succinic acid, which is really, really relevant in industry, pharma, food, Saccharomyces cerevisiae needs to expend ATP. This means that under anaerobic conditions, where your supply of ATP is very limited, you won't be able to do this. So, in our new strain, so this is wild type Saccharomyces cerevisiae. This strain just breaks down sucrose into glucose and fructose, and then in the end you have 4 ATPs per sucrose. Everybody can see this corner here? Prefer there? So wild type Saccharomyces cerevisiae produce 4 ATPs per sucrose under anaerobic conditions. Laser just failed. Really failed. Guys, we need another plan B, huh? Ah, depois ele apaga. So this is wild type yeast. By hand. Wild type yeast. So we increased the amount of ATP produced from the same amount of sucrose doing 2 modifications. We changed the way that Saccharomyces cerevisiae hydrolyze sucrose. So it's not hydrolyzing anymore. It is making phosphorylases. This means that the sugar is breaking down into 2 monosaccharides, but one of the monosaccharides will be already phosphorylated using inorganic phosphate, not using ATP. Doing so, you go from 4 to 5 ATPs per sucrose, which is 25% more energy for the cell. So now you can make some expensive compounds like succinic acid. The other modification is that this happens in the cytosol. So you need to bring sucrose inside. Wild type does this outside. But to do inside, you need to transport. Native transporters use energy, but we don't want to expend energy. So we took this transport from beans. It's a sucrose facilitator present in the coat of the beans, and we express this in Saccharomyces cerevisiae. Now, theoretically, we should have 5% increase in ATP. Wesley, do you measure ATP? We don't measure ATP. We measure biomass yield. Biomass yield and ATP yield is just the same thing. If the strain has more ATP, we'll produce more biomass. So we measure biomass yield in continuous cultivation in reactors, chemostats, and we got an increase of 8%. It's not 25, it's just 8%. But this is a significant proof of concept that the strategy is working. So we managed to publish actually 2 weeks ago in a nice journal. Thank you very much.
Before we finish this cycle of presentation pages, we have a very last one page from Carla Maia Brandão from Unesp.
Hi everyone, my name is Carla. I work with Professor Nelson Stradiotto from Unesp, Araraquara. And I'm the first year of the program. And my project is named Sustainability of Biogas from Sugarcane in Morro Agudo region. The general objective of this project is to study the sustainability of 3 applications of the biogas. The first one is the employment in agricultural machinery. The second is the incorporation in natural gas network. And the third one, the power generation. The choice of Morro Agudo region, the reason is because of the industrial pole that exists in this region, a lot of sugar mills. It is located near to the Brazil-Bolivia gas pipeline, and it's located near the main electricity consuming center, which can reduce the investment in power distribution. The relevance of this project, one of these is to reduce the Brazilian rainfall dependency, increasing the renewability of the Brazil's matrix. As you can see, when the year is dry, we have to drive the thermoelectric units that decrease the renewability of the matrix. So the biogas could improve this scenario. The second reason is related to the consumption of energy. As you can see, the industry and transportations represents almost 65% of the energy consumption, and the main sources of energy are natural gas, electricity, and diesel oil. These 3 can be replaced by biogas. We expected to deliver 3 business plans evaluating the 3 applications that I show you, and also make a qualitative analysis taking into account the social environmental aspects of sustainability. First, in the end of all the project, we expect to evaluate the reasons that the decision makers they consider to decide to invest or not. Because of this, I made that question to the professor. So is that. Thank you very much.
I would like to invite all the speakers to the stage. Carla, can you come to the stage as well? They were so good in keeping up their time that we have time for questions. So if you want, I please do encourage you to ask them. Anyone? Like 11 interesting works? Felipe has a question.
Hello. First of all, congratulations for all of you. It was a very nice presentation. Thank you for coming. And I have a question for Carlos. Carlos, I'm not sure did I misunderstood your name? Carlos? Sorry. Jorge Eduardo. Sorry. During your presentation, I didn't understand because you show us what's your challenge within your project, the problems that you are facing and you need to overcome in order to make this happen, your disposal. And I'm interested to know what is the advantage of using enzymes as catalysts within your project, because I understood what the gaps, but I want to know what is the good things about it.
The difference is conventional fuel cells use platinum-based catalysts. Platinum is very expensive because there is a little bit in the world. There is a depletion. It's a metal in depletion, the nature. And on the other hand, enzymes is a source of, is renewable. This is the principal advantage.
Okay. Thank you.
Okay, Renato has a question.
Hello, thank you. I look at Carla in the poster session, but I couldn't find her. So now I would like to know a little bit about the production of biogas in terms of sugarcane bagasse or in terms of tons of cane. Do you have this information about the production of biogas? Because biogas, considering in terms of chemical roots, can be used to reformation and to produce additional hydrogen production. And additionally, we have a huge market for hydrogen production. Do you have the information about the values of yield, conversion, everything that can help us?
In my project, the biogas will be produced from vinasse. Each liter of ethanol can produce 11 liters of vinasse. And the production of biogas will depend on the organic matter content from vinasse. So I have estimation, but I don't have the real numbers. The number is 0.3 normal cubic meters of biogas per gram of COD, the demand, the oxygen demand. Okay. So we have to do the estimation.
Yes, 0.3 versus 80 liters per ton of cane, around this calculation. Okay, yeah. Thank you.
You're welcome.
We have time for one more question. Okay.
Sergio, right? Thank you for the presentation. I'd like to know whether the engine industry are prepared for a B20 or higher than that.
It's a good question. Basically, our engine, or for not also our engine, but every engine produced for diesel engines can use it normally with biodiesel, B10, B20, B30, and so on. The problem is not the functionality of the engine. The problem is that we, for example, basically, let me say, the automakers basically produce the trucks and send these trucks for sales yard, okay? So during the sales yard, they have a time inside of this sales yard, around 1 month, for example, 2 months or 3 months, then deliver to the dealer for sale. Okay. Thinking about that, when we lost a window for exported window, then we need to wait more 4, 5, or 6 months there in the sales yard. Then we have a totally around near 1 year, around 1 year. So during this period, you start the oxidation reaction and increase this oxidative reaction. Then we start clogging, as a polymerizing the components start polymerizing and stop the functionality. For example, injector nose, the fuel system, and filter, and so on. If you use directly B20, for example, you put B20 and use it during 100 kilometers, 100,000 kilometers. Every day you're running, you have any problem. The problem is that is when you stop the vehicle and stay stopped for 1 week or 1 month, then the catalyst reaction starts, depend the situation, depend of sediment and waters, basically, increase the speed of the reaction. Then start the problem, yeah. But it's not regarding the volume of biodiesel.
So let's give another round of applause to our colleagues, please. Thank you.